My name is Crisalde Ramirez-Telis, and I am from the group 303. And my partner.
And you know me, we are very pleasure to have this work and show you how we know it. But proteins are a family for biochemical compounds. Their structure could be globular or fibrous. We know that detail because we have seen it before. These biomolecules, linear polymers, are built from about 20 different amino acids. The sequence of amino acids is essential since it determines the overall structure and function of a protein. A protein has several functions. It may serve as structural material, as a enzym, as transporter, as antibodies, or some other mechanism of expression.
Introduction. Proteins are biochemical compounds consisting of one or more polypeptides, typically folded into globular or fibrous form in a biological functional way. It is also recognized as the working molecules of the cell. It can be distinguished from fats and carbohydrates by containing nitrogen. Other compounds include carbon, hydrogen, oxygen, sulfur, and sometimes acid, and sometimes phosphorus. A protein is a linear polymer built from 20 different amino acids. The type and sequence of amino acids in a protein are specified by the DNA in the cell and that produces them. The sequence of amino acids is essential since it determines the overall structure and function of a protein. A protein has several functions. It may serve as a structural material, as enzymes, as transporters, as antibodies, or as regular. How do amino acids link together to make a protein? They link together by monomers. A protein is made up of amino acids. These amino acids are therefore polymers. These polymers are made up of are the ones that make the linking of the proteins.
These monomers are blocks of amino acids. They form in certain structures. There is a polypeptide structure, that's the name of the polymer. A chain of peptide bonds is considered the backbone of the structural framework of the protein. We can say that oligopeptide is the one who has minus 50 amino acids, and a protein who has most. You can see this is the structure of the polypeptide structure.
Polypeptide structures. As we can tell, yours are amino. Here was one of our amino acids, and the polypeptide, and this is the peptide bonds, which are the ones that are in blue. And on the last one, we see our carboxylate group. We have to know that this is our backbone. This is the structure that makes, and that at the end kind of makes the structure in globular.
How we can name this? What's the molecular structure? The primary is the sequence. We have already said what is the polypeptide structure. The secondary is the local folding. The proteins tend to fold. This means they need to do it, and sometimes it's spontaneous. We have the tertiary, there is a long range folding, quaternary or multimeric organization, and there is a supramolecular structure. It's a lot of scale of percentage. Now on, we will see this.
Okay, the primary structure is, as we have told you guys, every protein has a unique sequence or order of amino acids, which makes them function. There are 21 possible combinations of primary structures, and N equals the polypeptide length.
Secondary structure or folding is created by interaction.
Here we see it as individual polypeptides held together by some forces that hold together tertiary structures. It's more complex. And here we see a hemoglobin, a centromere, and we see how it's more complex than the secondary and the tertiary.
There is a protein folding. That's going to be telling you the interaction that is to do all the fibers and globular proteins. You can see in the first case, first step of the image, there is a ribonucleus protein that is being folded. It will fold to these groups, will be exposed and nuclei and will do their function.
Okay, protein folding. Some proteins fold spontaneously, and some proteins require help to fold correctly. The ones that both spontaneously or from the ones that can happen in our organism. And some that require help have some, like this one. There are two classes of critical and complex cell processes, synthesis of nucleic acids and protein signal transduction. Commentaries.
A fundamental goal of molecular cell biologists is to understand how cells carry out various processes essential for life. We as chemists, we need to know this already.
And more than a commentary, it's like a suggestion. Whether you are a vegetarian or a meat eater, you must have protein in your diet. The protein in the food we eat is our main source of chemical building blocks. We need to build our own protein molecules. Here are some of the proteins that we can get from fruits, vegetables, meats, foods containing fats, milk, dairy, and bread and cereals.
What we conclude? A protein is a very important structure in our organisms. There are two kinds of protein, primers and globular. Proteins are natural polymer molecules consisting of amino acid units.
We thank you for your given time, and any questions or any commentaries we would like anyone has, we would like to answer you.
Good. Do you have any question? No. Okay.
First one. Can you say me what kind of genes are present in the proteins?
What type of links?
Yeah.
The peptidic bone. They are the amino acids have the categories to have one monomer and other monomer. They have the peptidic bone between them. It's from the chains and it's from the groups. This was
Protein, for example,
the hydroxyl groups are very exposed during the folding. On the phosphorus, other iron. We have to see charges. You explain very well on the what the fuck? So that's not your doubt.
You know how is important. It's very important the protein. By example, do you know how many grams of protein do the human need every day? According to their weight. Okay? And say that the human body needs eat carbohydrates, but